Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Bacillus means little rod, and cereus means wax. So Bacillus cereus refers to a rod-shaped bacterium resembling a wax candle. This bacterium most commonly causes food poisoning, but it can also lead to more serious infections, particularly in immunocompromised individuals. B. cereus has a thick peptidoglycan cell wall, which absorbs purple dye during gram staining, identifying it as a gram-positive bacterium. It is also motile, equipped with thread-like flagella that enable it to swim in liquid media or swarm on solid surfaces like culture media. Additionally, it is aerobic, meaning it requires oxygen for growth. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. B. cereus is catalase positive, meaning it produces the enzyme catalase. This can be tested by adding a few drops of hydrogen peroxide to a bacterial colony. The catalase enzyme breaks down hydrogen peroxide into water and oxygen, causing the mixture to foam. B. cereus is also a beta-hemolytic bacterium, as its colonies cause beta hemolysis, or complete hemolysis, when grown on blood agar. This occurs because B. cereus produces a toxin called beta-hemolysin, which fully lyses the hemoglobin in red blood cells, changing the blood agar's color from red to transparent yellow around the colonies. Finally, Bacillus cereus is a spore-forming bacterium, capable of undergoing endosporulation when it senses environmental threats, such as extreme temperatures, dryness, or harmful radiation. During endosporulation, the bacterium first replicates its DNA, then forms a wall inside the cell, isolating a portion that we can call the mother cell. The cell's plasma membrane surrounds this mother cell and pinches it off, creating a separate entity known as a forespore. The forespore then invaginates into the mother cell and becomes fully engulfed by it. Inside the dying mother cell, the forespore loses water, accumulates calcium, and is encased in a super tough cortex derived from the mother cell. At this stage, the endospore can resist heat, harsh chemicals, digestive enzymes, and even antibiotics. Eventually, as the mother cell dies, the endospore is released into the environment. Remarkably, an endospore can survive for over 1,000 years, waiting for favorable conditions to germinate into the bacterial or vegetative form, which can then grow and divide. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. These endospores can also contaminate food like rice or pasta, and some of them germinate into vegetative bacterial cells that begin producing a toxin called cereulide within the food. Cereulide is a toxin known to cause vomiting when ingested, leading to an emetic type of food intoxication. Cereulide is heat-resistant, so even when food is cooked or reheated, the toxin remains. If someone consumes cereulide-contaminated food, the toxin binds to gastrointestinal mucosal chemoreceptors, excessively stimulating the vagus nerve, which triggers an exaggerated vomiting reflex. In addition to cereulide production, the endospores themselves can reach the small intestine, where they germinate into their vegetative form. Once there, they produce other toxins known as enterotoxins, which target the intestinal epithelium. There are 3 types of enterotoxins: hemolysin BL, non-hemolytic enterotoxin, and cytotoxin K. These enterotoxins damage the intestinal epithelium, leading to the secretion of electrolytes, such as sodium and potassium ions, along with water into the intestinal lumen, resulting in watery diarrhea. Finally, B. cereus can also contaminate medical devices, such as needles and prosthetic devices like central lines and indwelling catheters. From these devices, the bacteria can enter the bloodstream, leading to bacteremia. Once in the bloodstream, they can spread to other organs, causing infections, primarily in immunocompromised patients, such as those with an underlying malignancy or newborns with an immature immune system. If B. cereus spreads to the heart, it can cause endocarditis. If it reaches the bones, it may lead to osteomyelitis. And if it travels to the central nervous system, it can result in meningoencephalitis. Additionally, B. cereus can spread to the eyes, causing endophthalmitis if it invades the interior of the eye, or keratitis if it infects the cornea. Symptoms of B. cereus food intoxication vary depending on the type of infection. In the emetic type, nausea, vomiting, and abdominal cramps occur, typically beginning 1 to 5 hours after consuming cerealide contaminated food. In the diarrheal type, moderate to severe abdominal cramps are common, and symptoms usually start within 6 to 18 hours after ingesting the endospores. With bacteremia, symptoms include signs of systemic infection such as fever, chills, and malaise. In the case of endocarditis, additional symptoms may include night sweats and new cardiac murmurs. Osteomyelitis presents with bone pain and swelling, while meningoencephalitis is characterized by headache, confusion, and a stiff neck. Endophthalmitis can cause severe eye pain and swelling, and in some cases, the eye may bulge out of its socket, a condition known as proptosis. In keratitis, impaired vision may occur. Diagnosis of B. cereus food poisoning involves isolating the bacteria from vomitus or stool samples. B. cereus is confirmed only if there are more than 10 circumflex 5 colony-forming units per gram of the biological sample, or if more than 10 circumflex 5 colony-forming units per gram are isolated from potentially contaminated food, such as rice or pasta. Another method for detecting B. cereus involves testing for its toxins in the food using assays like latex agglutination tests, immunochromatography, and detecting the bacteria's genetic material using polymerase chain reaction, PCR. If a systemic infection is suspected, blood cultures are performed to identify the bacteria in the bloodstream. Treatment of B. cereus infections typically involves supportive care for foodborne illnesses. For invasive infections like bacteremia, the first step is to identify and remove or replace any suspected indwelling medical devices that may be the source of the infection. Following this, antibiotic therapy is initiated, with vancomycin being the drug of choice. Alternatively, imipenem and gentamicin can also be used. Summary of Bacillus cereus. To recap, Bacillus cereus is a rod-shaped, gram-positive, beta-hemolytic, catalyst-positive, spore-forming aerobic bacterium. It produces cerealide, a toxin that causes the emetic form of food intoxication, and 3 enterotoxins, hemolysin BL, non-hemolytic enterotoxin, and cytotoxin K, that are responsible for the diarrheal form of food intoxication. This bacterium can also contaminate medical devices, leading to bacteremia and various infections in immunocompromised individuals, such as endocarditis, osteomyelitis, meningoencephalitis, and eye infections. Diagnosis of B. cereus infections involves identifying the bacteria in biological samples or testing for its toxins using specific assays. Treatment generally includes supportive care for food intoxication, while systemic infections are treated with antibiotics like vancomycin. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you.